And I request Professor Anand Srivastava to take charge of this session and kindly steer us through. Here it is your. So first of
I'll invite Professor Biman Bagchi to come and give his talk on future ministry of medicinal. For those of you who don't know me, my initiation with Bagchi Lab started in 1998 when Shravani taught me chemistry first year. And so in some ways I am connected with this. So without much ado,
Thank you very much. Okay. So I said that some of the things that has happened in our group, which at that time you will think are out of pure curiosity. For example, but how did it happen? At one time I was exceedingly interested in the autoimmune diseases. And everybody says, at least in doctors or others says, autoimmune disease are because of lack of vitamin D. So I wanted to get into it, and with Shushmita and other students, we built a model, a network model, which some very similar to what you guys have seen yesterday or today. That thing. So it came completely from my real desire to understand the role of vitamin D. We have always gone into one, we are struggling to publish the second one. We have done even an immune response, which is a very complicated work. I won't go into this. And then I was interested why everywhere we say curcumin, healthy, and this great anti-oxidant and all these properties. What makes curcumin so interesting? So we did a work on curcumin, and I found one thing that probably I knew that the strained hydrophobicity and that drive and aggregation. Now, because I always knew that when you have it, when I used to climb trees, there was all these things, the butterfly, or what before that in Bengali called Shupoka. What is the English? I don't remember. Okay, that used to, when I used to climb trees, then I used to come back with full of those white stuff, which was very painful. Then somebody would take a blade and remove that and then put lots of haldi there. So that's a very anti-inflammation thing. And so I started, so that was one of my, again, doing the metformin came again from diabetes drug. Many of my friends take the glyphosate. And they are looking into the metformin, and it's such a simple, beautiful formula. Then we did a work on metformin and interaction with DNA. And insulin again came. And we are now doing several others, this kind of calculation. So what I tell my students in my class that in statistical mechanics, this is the advantage. I talk repeatedly of the chemotherapy track, anti-cancer in the class, which is what here, Almighty, and so otherwise several things on domomycin and actinomycin, ethidium bromide, and why they are so powerful as a chemotherapeutic drug. And you look at the DNA, and then you look, then we found recently with cobalt that some of these anti-cancer drugs are tremendously stable. When you go into DNA, they just don't come out. So we wanted to find out an equilibrium to do that. And some time ago we did dark DNA intercalation. So these are the kind of thing I call physical chemistry or medicinal research. And so physical chemistry teaches you with the fundamental principles. Along with statistical mechanics and quantum chemistry, you can really attack everyday problems. And the advantage of my young colleagues is that these are the kind of research which are not difficult to publish at all. We have no difficulty in publishing any of these things, and probably write few more. I do not have any difficulty to publish because people, the referees just love this work. I take 5-10 more minutes, that's all. So it's a what going on. Show mock, insulin, and metformin. She's insulin. She's not insulin. Biman Bagchi, Sushmita with curcumin and vitamin D. Anindo is the one we work with the metformin, Rudro, and I've not talked about other things. So insulin we deal with Professor Gopal, and these are metformin bunch. So there's a lot of these things going on, and we'll go on in the group. So I'll not be able to do, as I said, I'll take at most 10 more minutes. But it is essentially for my students in the class to tell that these are the real life things you or your father or your mother, somebody is taking your 50 or so you need a responsibility to understand these things. So you cannot just take a drug and put it in your body, okay? So then there is insulin is of course. So we looked into several beautiful stuff on insulin, which have given us big weight. CNN News covered it, and they went to Chicago. 2 professors just wanted to talk to me about insulin. Then I said metformin. We are supposed to do more work on metformin because I think there are a lot more to do. And I'll talk a little bit of curcumin, and then vitamin D is a huge project that Sushmita is going on. So insulin, it's the important thing about insulin is that it is a biologically active. Insulin is a monomer, but in a body it's stored as a hexamer. The hexamer goes over to dimer, and dimer then in the pancreas, it is stored as a hexamer. When it comes out, it becomes a dimer, and then the dimer has to break when need arises to become a monomer, because monomer is the active. So it started a very funny way. My youngest son Kushal was, I wanted to do some simulations, but then I found out that your son, whom you think is very hardworking, as lazy as your own student, other schools also, very interesting realization. Anyway, he came, spent a few days, and then he came out with the idea to do insulin, and Sushmita helped, and that was actually one of the very first papers from physical chemistry of insulin. I looked extensively, there are very few actually. Physical chemistry was, wow, what does it do, how does it do? But then, somehow we got into understanding hexamer, how hexamer is stable. And then Pooja started working on dimer A. So this is a series of what that. So this is the insulin. Monomer has 2 branches, A and B, chain A and chain B, and then the bend, the 3 disulfide bonds. This beautiful structure, helix otherwise, with a coil, beautiful structure, and that plays havoc in your body. Okay, and disulfide bonds. Monomer is a biologically active, not a very big protein, but just about the right size that one can handle. So this is the one Shomo Sayantar worked out, and we discovered that there are 10 water molecules. Very nice story. I like the number 10 because reminds me of the Agatha Christie famous 10 Little Indians. Why Agatha Christie had Indians, I don't know. I think Indians objected to it, then it became dogs, 10 little dogs. Famous story of Agatha Christie. So there are 10 little water molecules inside and 2 zinc, similar to the kind of things harmonic block or somewhat, and they are hexacoordinated. And we are really searching for the pathway how this hexamer breaks and how the hexamer becomes dimer. And we are playing around with its difficult things. That's one project going on. And so they're more stable than other. And this is the cell. You remove the water molecule, then it collapses in a very short time. But the pathway of breakage probably is different. Then, and I was really interested because see, those I know one of my colleague takes insulin and he is not supposed to drink or drink in moderation. So my idea was then, if I have ethanol, then the hexamer might not become dimer. Then you are absolutely in trouble. There could be other things also, but you need to have a starting hypothesis. So we looked at the interaction of insulin with ethanol. These are really difficult work, and they are the simulation and main drivers of their Shomok and Shanta. So this is the, many of these things are now quite well known work. So this is the detailed insulin net. So cavity water as flow, and they are the ones they weave and indicate hydrogen bond network among themselves, and that stabilizes. So the stability of hexamer surprisingly has this 10 water molecules inside, and ethanol destabilizes the and it makes it kind of a collapsed state. So really, if insulin go and ethanol goes inside, then it just does not. This process of going to hexamer to dimer and monomer, that's channel gates things. So this is then the insulin dimer, which recently came out, two other papers which then came out in VNAS, and this is a beautiful story which Pooja worked out, that how a dimer becomes a monomer, the insulin dimer dissociation in aqueous solution. Sayantal worked in the water and ethanol. This also we did the ethanol, and we found out the pathway. And there's some nice pictures. One got the Fianchy, and here again the kind of talk we have today, that we have ideas of doing some calculation done, but more going on. The pathways of going from here is the order parameters are number of native contacts in dimer. The two proteins are in touch with each other, and they have many contacts. The number of contacts is this one, and the radius. Separation between monomer center is R, mm. So as you separate the dimer becomes monomer, you go along this direction, and you also have to come down because contacts are broken. So this is a beautiful free surface. And Pooja found out, Pooja and Shanta, what are the things. So the one that people like a lot, the sequence of events we found out, and some of the free energy surfaces are very similar to free energy surfaces we saw in Sravani's talk. Now, this is the beauty actually, that how this water, and in the case of ethanol, how ethanol comes in the hydrophobic and keeps them together. Now you cannot easily break. That's what I was saying. Ethanol screws. This is a very essential process. Okay, and so this is another point, that when you separate them out, water comes from the beginning. This is another talk. You have to make the water comes in, and this beautiful picture, that when they are even center to center distance is so many, 50 angstrom apart. So surface to surface is 30 angstrom, 20 angstrom, something like that. You have thousands of water molecules. But these water molecules inside, even at such separation, is so different from water molecules in the bulk. There are a lot of fluctuations between water and water studied by institutions. Now, I'll just say this, I said, why
Professor, please go back. I want to give the conclusions on insulin. Okay, go ahead, please.
There are actually, this is the ethanol part, then there is the real part. I'll come and discuss with you. I'll tell you all these things. I have been begging Yashona to write a paper together, but Yashona doesn't want to write paper with anybody. So,
sir, can I ask?
yeah.
This insulin that you get in the market, particularly these rapid acting insulins, they are always stored in viscera. And the one is there so that they could stabilize the monomer form. So once you take it out of the body, so that acts fast. You don't wait for this conversion from
Right.
That's
a new insulin.
That this ethanol probably that will also go for viscera.
Yeah, the way it started, my project started after the shots, early initial paper. It started by the Shomok Shant, and you should cite that paper in your papers. You never cite that. Was that I wanted to do the aggregation because this is the aggregation. So I talked with Peter and and both of them said, Biman, there is money in this project. That means you can swindle some money out of pharmaceutical companies. Because not only they are aggregates, you cannot store insulin too long in the bodies. And that's a huge pharmaceutical problem. That was where I started first. And then all these things are going on. So this beautiful molecule metformin, this is the number one or perhaps the only drug of diabetes, type 2 diabetes. So look at the beauty of this molecule. As a chemist, you have to appreciate that, they are all nitrogen and there are few carbon, but how many nitrogen are there? 1, 2, 3, 4, 5. So we want to know why it has so many, what is the reason for the activity? So we studied its structure, we developed the force field, and we did the by the way, these papers, this kind of papers, when they go with the reference, just don't have any comments. It's like the initial paper only one. We go, remember only they are just ecstatic about what it is. Okay, so protonated form is used to avoid as this glycophage, and so we wanted to see what happened when you, it has anti-cancer property that we started to work. So Shweta remembers about that now. No, we didn't go anywhere, so you should go back to that. Then we did the force field. We looked into that there is a Brenner form. We calculated the diffusion coefficient, and the Brenner form is the one which we thought is the that goes into DNA. So we looked into the DNA interaction between DNA and metformin because it's also acts as anti-cancer drug. Though I think it's not because of that, because it sits in certain receptors. But we worked a lot of things and how it binds, how metformin goes, and so this is a metformin little thing. So this now the curcumin, the whole thing. This is again the first paper. Metformin is the first paper. Haldi is the first paper. Then we know what paper existed before this. So now this is another beauty, and these are really in love with this kind of problem now. That ketoenol tautomerism, and then all these different curcumins, how they function, why they function. So as a physical chemist, what can you do? You of course understand the structure and its amount of dynamics, but that's not too important. Because one thing was convinced that the reason curcumin or Haldi is so important because it has a hydrophobic interaction. And yeah, I can decrease inflammation in any place on my leg if I put Haldi and chew, because they will take the water out. And if you just dry the place, then inflammation and infection dies. Okay. This was my initial thing. And that indeed happened. So we, in disease we use it. So we just put, and then when you put several curcumin together, then there is a beautiful self-assembly where they dry water out. So we could go, and there is a nanoparticle, nano assembly that is so popular, and different kinds of way that is the radial distribution function you can find from the center, and then we did up to 16, and it just then we study the character of the that what kind of structure it forms, cloth rate or inverted cloth rate of hydration, how it interacts. It's tremendously slower than bulk water, but this whole thing becomes very stable, exceedingly stable. And then create a landscape, once you get the beautiful structures like this one, you can find the creative landscape. Milan and Sushmita did enormous amount of work on this, and I was once invited to give a talk in a conference in Sri Ravishankar. I went and gave the talk, and I of course had great fun talking to those people, Ayurvedic people, and all the stories I told of from Putunachini Kora, story of dancing dolls of my number one probably East province. And there this great guy Yadav, Ayurvedic doctor, tells Soshi, the protagonist, that what does your medicine talks about this Surya Vigyan, the science of sun. So she said, no, it says nothing. Then the famous line of this old Ayurvedic doctor, how can you treat a person when you don't have the very initial Adi Gyan? That means first thing you don't have. How can you treat people? And that was what actually, when I told there was an excitement in the audience, some of people went out and come. Then says Sri Sri, he wants to see. I said, no, I really have no time to go back. Then they preponed the thing and took me to Sri Sri Ravi Shankara. It was amazing. I am really very grateful that I got to see the great man. But he gave me vibhuti and sort of nice things, all because of Boldi and okay. So I had vitamin D. Vitamin D, fish and all these things. And some of you don't like fish, but if you better take fish, you get vitamin D. And these are another beautiful motive. So initial things which Shushmita and myself started trying was with the steroids. Huge effect of steroids in human body, and steroids can kill you. Vitamin D is very much like a steroid. So we wanted to understand how that works. How does it, so it's a vitamin D receptor which accepts vitamin D, and then it synthesizes 980 enzymes, but it plays a very important role in protecting you from all kinds of pathogens. So we build a very big T cell regulation. This is a killer cell. There are two killer cells, one a regulatory killer cell and the effector T cell. Effector T cell kills the pathogen, but regulator T cell has to come and decrease the effect of the. And there's a beautiful stem cell kind of thing that going on here. And then we formed the network model, and then that was this chemical network model. This scary picture that shows me the thing. And this is the network model, just like many of you showed. And I call it systems viability. And that we found out the bistability, dynamic bistability. So this is the paper, main paper. And then we can strong regulate it. It is a huge story, but we introduced one thing which is very nice. We introduced what is an immune response function in terms of fluctuations in the T cell. That's a new thing, but that's why they are having difficulty with publishing, but we don't really care too much, we will put it in format. But so we describe the immune response functions. And so take home message is that vitamin D preserving bistability phase diagram of immune regulation. We found there is an optimal T cell, which of course no, but it comes out of the model and the theory. And I think, thank you very much for your attention. So, I don't want to talk at all, but one thing I must tell, that the problems actually need not be, they are there. Life is getting more complex. And so you should be look at that way also. Okay, thank you.
Like there are hundreds of people. If you go to an IISc website, there are hundreds of them.
Yeah, website. Yeah, look for like Serenamity and Saturday.
So
You have psoriasis, you have rheumatoid arthritis, yeah, huge number. And it was covered, our work was covered in newspaper because one person came from the Canada.
So is vitamin D responsible for all of them, or there are other things also?
We don't know really. Isn't it? It sometimes drives you mad that people are not doing these kind of problems. Again, our work is only work. So, it's a complex process. Even this ugly thing that usually that cookie monster kind of thing. So, even that has so many steps, and it is a highly minimalistic model, course-led model. And we need a lot of experimental results. I went and talked to doctors that we need to see see, we are taking all this time, blood, and we are getting the T-cell. That's one thing that comes out in your blood report. And I think it's not that difficult to calculate the fluctuation in the T-cell. And if your fluctuation is healthy, you are okay. But if I find your fluctuation has gone down sharply, then you are in trouble because your immunity is down. So we have delta T square as an immune response, we call it IMI, immune response function. And it is extremely important to have a lot of fluctuations in your body, just like specificity and compressibility is also. That is, I'm saying I can tell Amananda Chandra has this immunity with respect to this thing, then that will really help you to protect yourself. But it's a long shot. A lot of work to be done. Here we work almost alone, one student once.
Is it not that curcumin is in the same family as sunset yoga? No, the reason I am asking that this molecule has a different assembly pathway. Because they are supposed to be a class of molecule called isodesmic assembly, meaning they are when the free energy as a function of is a constant increment. They don't have any free energy minimum.
Absolutely, absolutely same here. Absolutely. You go on adding more curcumin, it goes to bigger assembly. Yeah, there is no size. Yeah, it's a downhill. It's a downhill.
Is that
It's a downhill.
But then in your
It is not like no, it is not like your. We have done up to 16.
yeah.
It is not like what we did with this triangle. What is the dendrimer? Dendrimer, it stops, right?
Six percent is not isodesmic, non-isodesmic.
Yeah, right. But it ultimately, beyond a point, you cannot grow it.
Yeah, there will be free energy.
Yeah, right.
With the
Yeah, right.
specific
Because there is no place. But here, in this three-dimensional thing, so there is, we have not found what limits the growth. Yes, if it has to limit it, then its own, it will be such that it has to have chains which are strongly hydrogen bonded, so that no other, with those phenyl kind of rings, actually cannot come and secure it. We have not studied this part also. There is one paper on you, Lord. And we should go back to this, but we have not done. But it's getting well cited, our vitamin D paper, first citation was Journal of Diabetes, then Pediatrics, that kind of thing.
But still at the equilibration time, we found like among 16, there is that is basically the critical nucleation size that we could reach. And two kind of morphologies we could found. One is parallel morphologies, where parallelly they are stacking, and another is anti-parallel stacking. So these are basically the sort of...
Yeah, very much like your
here the people probably won't
very much
in
like
the same class, then people will talk about what's it called J aggregate or H aggregate instead of correct. So
it's like Alzheimer proteins,
Yeah, which one is-
like Alzheimer proteins, parallel, anti-parallel, that kind of things. Yeah.
Is a diabetic not supposed to drink, according to you?
Absolutely. You are not. Then and that's the first thing a doctor tells you. If you become diabetes, the biggest loss is that you cannot drink. And it doesn't mean, since you don't drink, doesn't mean that you will not have diabetes.
Yeah. So, sir, Krishna wants to say a few things.
Yeah.
Excellent and wonderful talk,
Thank you.
especially at my age. I'm not ashamed of to say that I'm a diabetic for the last 15 years. It's a lifestyle disorder, and keep on taking medicine after medicine, dosage keep on increasing. Therefore, I'm very happy to listen to some of the physical chemistry based fundamental aspects of both insulin and metformin.
And the problem is that kidney membrane, and you have these helices, which are transporters. Problem is that as we grow older, same happens in our brain everywhere. Some of these helices and some of the proteins get stuck in our body as junk, and they don't go out anymore. But they don't allow function at the same time. So, what I'm trying to say, everywhere in medical research, there is an intellectual component which is not being brought in. And physical chemistry can give you that intellectual component and try to think little bit why my kidney will go, you know, because if I eat dopamine, then what will happen? So all these little molecules, see, it is chemistry because these are molecules that made for me. Don't expect a physicist to do it. We have to do it. And but then physical chemistry, quantum and statement together, the wonderful area of research. That's what I wanted to tell
Wonderful
my
area for
students.
Indian people.
Yeah.
Because lots of things have to be understood and looked for.
No, thank you very much for guiding us. We are late. So I'm done.